Hello, my name is Alexis Wilson, and today I'm going to talk to you about ATAC Seq for my local expert presentation. So ATAC Seq stands for Assay for Transposase Accessible Chromatin with High Throughput Sequencing. This assay uses the hyperactive transposase TN5 to insert sequencing adapters into open chromatin regions. This allows researchers to gather information about chromatin accessibility across the genome, regardless of the mechanism responsible. So quickly, why am I talking about this? Well, it relates to our module on next generation sequencing. See, ATAC Seq specifically relies on next generation sequencing library construction using TN5. The library that is then generated can be sequenced by NGS, and the regions of the genome with open or accessible chromatin are analyzed using bioinformatics. ATAC Seq is a two-step process. The first step is going to be cell preparation. The cells need to be harvested and then counted out, as the cell count is super important for this assay. The cells must also be intact and in a homogeneous single-cell suspension. Then they are lysed with non-ionic detergent to yield pure nuclei. The next step we have is the transposition reaction. Here the chromatin is fragmented and tagmented with sequencing adapters simultaneously using TN5 transposase to generate the library. Amplification using PCR is performed after this, and analysis of the resulting library can be performed by qPCR or NGS. For this assay, usually there isn't any controls due to the cost, but if there were, it would likely be genomic DNA from the sample that is fragmented, usually by sonication, has adapters ligated, and is sequenced along with the ATAC sample. One application of ATAC Seq was performed by researchers in Miami. They wanted to investigate the effect of the polycomb repressive complex 1, or the PRC1, on chromatin states in breast cancer. The ATAC Seq experiments showed that depletion of the ring 1b, a member of the PRC1 complex, leads to a change in chromatin accessibility at enhancer regions, as you can see by the figure down below. Loss of the ring 1b led to the loss of about 500 accessible chromatin region peaks and the appearance of more than 600 new open regions near enhancers. Overall, these results confirm that the ring 1b has dual function in regulating transcriptional programs in breast cancer cells and does so by altering chromatin accessibility for key transcription factors and chromatin organization proteins. ATAC Seq analysis also revealed a widespread decrease of chromatin accessibility and age-related macular degeneration by scientists at Johns Hopkins University School of Medicine. In part B of this figure, it shows the instances of open chromatin in the retina and RPE from healthy controls, NORs, and AMD patients. Further analysis of this data then showed that specific transcription factors and cellular functions were enriched in differentially accessible chromatin regions. We also had researchers at Cornell that used ATAC Seq to study the open chromatin regions in sorted viable germinal center B cells from LSD1 mutant mice. They observed 733 regions that gained chromatin accessibility upon LSD1 knockout, and 314 regions lost accessibility. There was also an increased transcriptional activation associated with those 733 opened regions. Further experiments also revealed that the BCL6 directly binds to LSD1 protein and recruits it to chromatin, and this interaction is required to drive the malignant transformation of germinal center B cells. The main advantage of ATAC Seq is going to be that there's a lower number of cells that are required for this assay compared to other techniques such as FAIR-seq and DNA-seq. Also, the simplicity of this assay, since it's only a two-step process, it can be done in several hours versus the other previously done methods that can take far longer. And it also can be used to generate a high-resolution map of nucleosome positions as well as transcription factor binding profiles. I would say the main disadvantages here is also the cell amount that they have to use. It's a very specific 25,000 to 75,000, and using too few or too many of the cells can lead to over or under digestion, which will lower the overall quality of the library. There's also a well-known issue with ATAC Seq that there is a lot of mitochondrial DNA in the data sets, and they've actually come up with a potential solution of ATAC method, which uses a detergent to remove mitochondria from the samples prior to the sequencing. Below are the list of the citations of the articles that I've used. Thank you for listening to my presentation, and let me know if you have any questions, comments, or concerns. Thanks.